DNA is made up of three things: deoxyribose sugars, phosphate groups, and nitrogenous bases. An important feature is that it is anti-parallel, so these two strands right here run in opposite directions. There are two kinds of nitrogenous bases: purines, which have two rings and will be your adenine and your guanines, and pyrimidines, which have one ring and will be your cytosines and your thymines in DNA, and uracil in RNA. In the case of base pairing, adenine bonds with thymine through two hydrogen bonds right here, and cytosine bonds with guanine with three hydrogen bonds. And these hydrogen bonds that connect the nitrogenous bases are relatively weak and can be broken by heat. So a key component of DNA are deoxyribose sugar molecules, shown in pink right over here, which are connected to negatively charged phosphate groups by phosphodiester bonds. And these bonds are hard to break, and if you take a closer look here at a ribose molecule and a deoxyribose molecule found in DNA, then you can see that you have 5 carbons, and so a phosphodiester bond links at the 5' carbon right here and the 3' carbon right here. At the 2' carbon right here, you are able to differentiate DNA from RNA if the sugar lacks one oxygen right here, as you can see. And right here at the 1' carbon is where the nitrogenous base links. With major and minor grooves, we'll be taking a closer look at the sugar phosphate backbone here. So in the spiral of the DNA, the major groove is the part where the backbones are farther apart, and then the minor groove is where they are closer together. Another important feature is base stacking, and you can kind of see it here where they are just stacked on top of one of the other. And this can be explained by nitrogenous bases being more hydrophobic, which means that they repel water, and the backbone, so the phospho-sugar backbone, being hydrophilic, which means that it attracts water. And so because the bases are hydrophobic, through hydrophobic interaction, the bases will want to attach to each other, and this will be amplified through stacking as they are enclosed by the backbone, and that will stabilize the DNA, double helix structure of DNA. Usually DNA is right-handed, where the direction of the helix goes up right here and then down like this. However, in these 5 examples I have, the DNA is left-handed. And a short little trick that I learned to differentiate this is that right-handed DNA will follow your left hand, and the backbone will follow this kind of groove, and then left-handed DNA will follow your right hand with this kind of groove. And so using that little trick, I found all these examples of left-handed DNA, and also by comparing with my right-handed DNA here.